So we have already studied about cellular adaptation. The next topic is reversible cell injury. See, the most common cause of cell injury is hypoxia. Hypoxia refers to reduced oxygen supply to tissue. And the most common cause of hypoxia is ischemia. Ischemia refers to reduced blood supply to tissue. And when there is reduced blood supply to tissue, obviously there is reduced oxygen supply to tissue because it is your blood that carries oxygen to tissue. Since we are going to learn about the various cellular and morphological changes that take place in a cell going through reversible cell injury, let's take the example of the most common cause of cell injury itself, and that is hypoxia. So what I'm drawing here is an artery. Not just any artery, but the renal artery that carries blood to your kidneys. Within this renal artery, there is formation of a fibrofatty plaque due to accumulation of fat. So there is accumulation of fat within the artery resulting in formation of a fibrofatty plaque, due to which renal artery stenosis or narrowing of the renal artery takes place. So if there is narrowing of the renal artery, obviously less blood will pass through it. Less blood will pass through the renal artery. So there is a reduction in blood supply to kidney. And this is referred to as reduction in blood supply to any tissue is referred to as ischemia. Since there is reduced blood supply, there is obviously reduced oxygen supply as well. And this is referred to as hypoxia. And hypoxia leads to cell injury. The very first change which is seen in cell injury is mitochondrial dysfunction. And it makes complete sense. Here's why. See, there is dysfunction with mitochondria. What is the function of mitochondria? Mitochondria is responsible for the synthesis of ATP, and it does so after oxidative phosphorylation takes place. And for oxidative phosphorylation to take place, oxygen is required. But since there is hypoxia, there is reduced oxygen supply to cell. So there is reduced oxidative phosphorylation within the mitochondria of the cell. So there is reduced production of ATP by mitochondria. That is, mitochondria is unable to produce as much ATP as it needs to. So there is problem with the function of the mitochondria or impairment in the function of the mitochondria. So mitochondrial dysfunction takes place. So what is the very first change which is seen in cellular injuries takes place in cellular injury? It is the mitochondrial dysfunction. Why does mitochondrial dysfunction takes place? Impairment in the function of mitochondria, that is production of ATP by mitochondria, takes place due to reduced oxygen supply to the cell. So what is happening? There is reduced oxygen supply to the cell. So there is reduced oxidative phosphorylation within the mitochondria of the cell. So there is a reduced production of ATP by mitochondria, referred to as mitochondrial dysfunction. When there is reduced ATP synthesis within the cell, changes at 3 different levels take place. Okay, changes at 3 different levels take place. Number 1, at the level of plasma membrane. Number 2, at the level of cytoplasm. And number 3, at the level of nucleus. Let's look at each of the changes. First, at the level of plasma membrane. So let's say this right here is a normal cell. In the plasma membrane of this normal cell, there are 2 pumps, very important pumps that are present. First pump pushes sodium outside and pulls potassium inside, referred to as sodium-potassium ATPase. How to remember? Remember the word pumpkin. So it's such a pump that pulls potassium in. How to remember the number of ions it pulls in and pushes out? See, since sodium has 1, 2, 3 characters and potassium has 1, 2 characters, it pushes 3 sodium out and pulls 2 potassium in. The second pump, it pushes calcium outside. So this is referred to as the calcium pump. Both sodium potassium ATPase and calcium pump, they are ATP dependent. That is, they utilize the energy from the ATP to carry out the pulling and pushing of the ion to maintain ionic equilibrium. But the problem is there is no longer ATP synthesis taking place. There is not enough ATP for the cell to utilize. So the pumps, they are unable to work. They do not function. They are unable to work. They are unable to push sodium outside and calcium outside. As a result, within the cell, accumulation of two ions take place. Number one, accumulation of sodium ions take place because sodium potassium pump is unable to push it outside. And number two, accumulation of calcium ions take place because the calcium pump is unable to push it outside. Now, due to accumulation of sodium ion and due to accumulation of calcium ions, two different important changes within the cell take place. Let's see what are they. We know this much, that water follows sodium. Wherever sodium goes, water also goes there. Since there is increase in the level of sodium within the cell, water enters the cell as well. We know that calcium is responsible for the activation of cellular enzyme. And since there is accumulation of calcium within the cell, there is activation of various cellular enzyme. Most importantly, phospholipase. So let's look at what happens when water from outside accumulates inside the cell, number one. And number two, what happens due to activation of phospholipase by calcium ion. First, due to accumulation of water within the cell obviously swells up. When water goes inside the cell and accumulates inside the cell is gonna fatten, the cell is gonna swell up. And this is the very first morphological, I repeat, morphological change that happens in cell injury. What is the first morphological change in cell injury? It is cellular swelling. Why is cellular swelling taking place? It is taking place due to the accumulation of water within the cell. That is why the change is also referred to as hydropic change. Okay. So now it is not just the cell that fattens up. The cell organelles, within them also water accumulates and they fatten up. For example, endoplasmic reticulum. Endoplasmic reticulum also swells up. What if we have such a cell which has microvilli? When accumulation of water within the cell with microvilli takes place, obviously the microvilli disappears. Microvilli disappears. Why? Because the side with the microvilli stretches. It stretches and the microvilli gets lost. Third, this water accumulated within the cell eventually pushes on the plasma membrane even more. As a result, bulges are created at different points on the plasma membrane. Bulges are created. And these bulges that are created like these are referred to as blebs. And the process is referred to as blebing. So blebing takes place. So what has happened? Due to accumulation of sodium within the cell, there is accumulation of water within the cell as well. Due to accumulation of water within the cell, the very first morphological change of cell injury takes place. That is cellular swelling, also referred to as hydropic change. It is not just the cell that swells up, even the organelle within the cell like endoplasmic reticulum that swells up. If the cell has microvilli, due to the swelling of the cell, the microvilli disappears. And at various points in the plasma membrane of the cell, bulging appears. This is referred to as blebing or blebs. Second, what about phospholipase? Now, this phospholipase that has activated, it has A's on the end. That means it's an enzyme. It breaks phospholipid. Where is phospholipid? It is found in the membrane of membrane-bound organelle. Once the phospholipid gets broken down, it curls itself. Okay, first the phospholipid is broken down. Now this broken down phospholipid, it curls within itself. See, phospholipid contains phosphate. Since phospholipid is broken down, now the phosphate becomes free. And the phosphate, they are negatively charged. These negatively charged phosphate, they attract the positively charged calcium ions, intracellular calcium ions. What we have here at the end is broken down phospholipid plus calcium ion. How are they present? They are present in the form of concentric lamellated curls, and this is referred to as myelin figures. So during reversible cell injury, there is formation of myelin figures. That was the first change at the level of plasma membrane. The second change is at the level of cytoplasm. Let's see the second change. So I said that it is not just the cell that swells up, even the endoplasmic reticulum present within the cell swells up. There is swelling of endoplasmic reticulum. Due to the swelling of endoplasmic reticulum, the ribosomes that are attached on the surface of rough endoplasmic reticulum gets detached because it gets pushed off the membrane. So the ribosomes, due to the swelling of the endoplasmic reticulum, gets detached. Detachment of ribosomes take place. Ribosomes are responsible for protein synthesis, but now since the ribosomes have detached off the endoplasmic reticulum, protein synthesis does not take place. That means synthesis of apoprotein does not take place. Apoprotein is responsible for the formation of lipoprotein, and lipoprotein is responsible for mobilization of fat. But since protein synthesis does not take place, apoprotein synthesis does not take place. Since apoprotein synthesis does not take place, lipoprotein synthesis does not take place. Since lipoprotein synthesis does not take place, mobilization of fat does not take place. So where will all this fat like cholesterol go? They will accumulate within the cell, and this is referred to as fatty chains. So when water accumulates within the cell, we call it hydropic chains. When fat like cholesterol accumulates within the cell, we call it fatty chains. Okay, third is at the level of nucleus. Since there is no oxygen supply, an aerobic respiration within the cell does not take place. Instead, anaerobic respiration takes place for the production of ATP. But when there is anaerobic respiration, there is formation of lactic acid. And lactic acid accumulation results in decrease in pH of the cytoplasm and increase of H plus ion within the cell. Now what this H plus ion does is that H plus ion, it attaches with the histone proteins. What are histone proteins? So histone proteins are present in number of 4, and they wrap around the chromatin material of the DNA and pack them together. These H plus ions, they go and attach with the histone protein and then bring these histone protein close to one another. The histone proteins, they are brought, there's 4, there's another 4, and the histone proteins brought close to one another, and so is the chromatin fiber. It is also brought close to one another. As a result, nuclear chromatin clumping takes place. Even when you look within the cell, within the nucleus of the cell, you can look at these clump formation of the nuclear chromatin material. That is all because the chromatin fibers are brought close to one another by the H plus ion generated due to the accumulation of lactic acid by anaerobic respiration. Okay. What is the very first morphological change of cell injury? I repeat, the very first change of cell injury is mitochondrial dysfunction. But the very first morphological change of cell injury is hydropic change or cellular swelling. See, this is the parenchyma of the kidney where reversible cell injury has taken place. So you can see that within the tubules of the kidney, there are cells. And these cells, they have swollen up due to the accumulation of water in them, referred to as hydropic chains. Everything that I have taught till now under the topic of reversible cell injury has been mentioned with the help of a very nice flowchart in Robbins. This is the exact flowchart from Robbins. See, so first there is ischemia, which results in less blood supply to tissue, which results in less oxygen supply to tissue, which results in hypoxia, which is the most common cause of cell injury. When there is hypoxia, cell injury takes place, and the very first change that is seen is mitochondrial dysfunction. That is, there is impairment in the function of the mitochondria because it is unable to produce ATP. See, if there is reduced oxygen supply, there is reduced oxidative phosphorylation within the mitochondria, and there is obviously reduced production of ATP by the mitochondria. When there is reduced ATP production, changes at 3 different levels take place. Number 1 at the level of plasma membrane, number 2 at the level of cytoplasm, and number 3 at the level of nucleus. Number 1 at the level of plasma membrane, the pumps that utilizes ATP no longer function because there is no ATP. These pumps are sodium potassium ATPase and calcium pump. As a result, there is accumulation of calcium and sodium within the cell. Sodium pulls water in, so there is accumulation of water within the cell as well. Now, due to the accumulation of water within the cell, the very first morphological change of cell injury takes place, and that is cellular swelling, also referred to as hydropic change. It's not just the cell that swells up, even the cell organelle like endoplasmic reticulum swells up. After that, if the cell has microvilli, due to the swelling of the cell, the microvilli is lost as well. At certain points in the plasma membrane, there is formation of blebs like these. Blebbing takes place. Okay, I said that endoplasmic reticulum gets swollen as well, due to which the ribosomes attached on the wall of the endoplasmic reticulum gets detached, it gets pushed off. Ribosomes are responsible for protein synthesis. Since ribosomes are no longer there attached with endoplasmic reticulum, protein synthesis does not take place. Instead, accumulation of fat within the cell takes place, referred to as fatty chains. When water accumulates, hydropic chains. When fat accumulates, fatty chains. Since there is no oxygen, aerobic respiration does not take place. So in order to produce ATP, anaerobic respiration takes place instead. But anaerobic respiration results in the formation of lactic acid. Due to the accumulation of acid, pH within the cytoplasm falls and H plus ion increases. H plus ions are responsible for clumping of nuclear chromatin material.